So thank you very much. Thank you very much for the invitation. It's wonderful to be here. It's wonderful to see so many faces and not just look at my screen. And it's wonderful to see how much structural biology can contribute to maybe avoiding these kinds of situations in the future. Now, what I would like to tell you about is another, I hope, complementary technique that I think can give you information, especially from the side of dynamics and molecular mechanisms in macromolecular systems. And so what I would like to talk about today is protein interactions. And most of you, when we think about protein interactions, probably think about pictures like this. Beautiful structures that we now have from decades of successful structural biology. We can go in, count every hydrogen bond, every salt bridge, and learn an enormous amount about the structural properties of these systems and how they interact. But of course, we've also known for a few decades now that there's a group of proteins that is a little more difficult to address with the same kinds of concepts. And these are intrinsically disordered proteins. And just to show you one of the famous examples, the tumor suppressor p53, that does contain a folded part that interacts here with the DNA, for instance, but it also has these large disordered regions. And current estimates are that 30% or probably more of all proteins in the human proteome, for instance, are intrinsically disordered, which means they're either completely disordered or contain large disordered regions. So it's not just a few proteins, it's many. And the other point I want to make is that many of these proteins are involved in interactions. And so again, if we take this famous example of p53, this protein alone binds to more than 100 cellular targets via its disordered regions. And so really paying attention to how these disordered regions interact is something that we think is worth doing. And so the way we do this is using single molecule spectroscopy, and since many of you may not be familiar with this technique, let me mention briefly, or let me tell you briefly how it works. And so what we do is we have our molecules either immobilized on the surface or freely diffusing in the solution, and we label them with a donor and an acceptor dye, and then what we can measure in this way is a quantity that we call the transfer efficiency in the simplest case, just by counting the numbers of photons that we get from the acceptor and the donor, and then this transfer efficiency, and that's what makes the technique interesting, is something we can relate quantitatively to distance information between the two. And so we have this distance dependence of the transfer efficiency, and so we can relate it to little r here, the distance between the two fluorophores. And that's why Louis Stryer called this technique a spectroscopic ruler, because we can measure distances on a nanometer scale, but we cannot just measure distances, we can measure dynamics, and this is something I would like to emphasize today. Now it turns out this is not the only thing we measure, but with these kinds of instrumentation that we use, which we call multi-parameter fluorescence detection, we not only get this wavelength-specific information about the color of the photons that we need to measure these transfer efficiencies, but we also do time-correlated single photon counting, so we know every photon has arrived within a certain time. The time resolution of that is in the picosecond range, so we can use that to get information about fluorescence lifetimes, about anisotropes. We can do correlation spectroscopy. I'll just show you a few of these facets and how we can use them to investigate proteins. Now, one of the key strengths of single molecule spectroscopy is that it can address a broad range of timescales, and as you are all aware, dynamics and biomolecular systems cover a huge range of timescales. From nanosecond dynamics of, say, disordered chains and how they reorganize, all the way to the very slow processes that we would have in protein aggregation, which in the extreme case can occur on the timescale of human lifespan. So there's a broad range of timescales, and over the years, the community in single molecule spectroscopy has developed a lot of complementary techniques that use this information from single molecule experiments to cover a very broad range of timescales. And I will not have time to go into the details, let me just highlight a few aspects that may be of interest. So on the short time scales, for instance, the fastest times that we typically probe in our experiments is with this method that we call nanosecond fluorescence correlation spectroscopy. So what we can do here is we measure distance fluctuations between the fluorophores. We attach the two dyes to our intrinsically disordered protein. This way we can measure how quickly such chains change their configuration, how they reconfigure. And often what we see for IDPs is this component here in the correlation functions on the time scale of about 100 nanoseconds. So this is the typical time scale of reconfiguration in IDPs, very fast, sub-microsecond, long-range motion. So this is sort of on the shortest range that we can address. Then, for instance, we can get information about non-equilibrium kinetics by combining it with rapid microfluidic mixing. We can cover timescales from milliseconds to minutes. And then there's this whole class of experiments where we can immobilize the molecules on the surface and then we can look at them for many seconds or minutes. And so, for instance, the resolution isn't, I hope you can still see these little blimps here in this trajectory. So every little signal change you see here corresponds to a signal binding and then a dissociation event. And so you can characterize the kinetics of such systems on a single molecule basis. And I would just like to use this to show you that we can cover a broad range of timescales. And quite a few of these experiments are now starting to become possible also in live cells. And so this is, of course, one of the next big challenges is to link the behavior that we see in vitro with the cellular behavior. But now I'd like to focus on a particular system of starting with two proteins that we investigated. It all started with this protein here by the name of Prothymosin alpha. This is a full-length human IEP, only about 100 residues long, and as you can see here in the sequence, it's full of negatively charged residues. And the result of that is that it has a net charge of -44. And from a lot of work that we and others did over the years, we know this protein pretty well. There's no significant structure formation under any conditions that we know of. You can imagine a protein that essentially lacks hydrophobic residues that would allow packing of a core. It's maybe not surprising. Such a highly charged protein always remains disordered, even when you inject it into cells. It remains disordered and as dynamic as in buffer. But then the question is, what does it actually do? So we wanted to know more about the interactions of this protein with other proteins, and there's a number of interaction partners that have been characterized over the years. And one of them that struck us as a particularly interesting case is this histone H1. Histone H1 is maybe a protein that you are more familiar with than Prothymosin alpha. This is a protein that binds not here at the core of the nucleosome where you have all of the core histones, but it binds here on top of the nucleosome on the diad axis. And there's this little globular domain that's also indicated down here that binds here to the DNA, and then it has these long disordered regions, and especially from single molecule experiments that I'll not have the time to tell you about, we know that this disordered part of H1 even remains disordered when it's bound to the nucleosome. And so when H1 binds to the nucleosome, what happens is on a small scale that these linker DNA arms sort of close up when H1 binds. So this would be on the level of the nucleosome. And on the chromatin level, H1 leads to condensation of chromatin. So it's a very important overall regulator of transcription in the nucleus. But what's more important, interesting from a structural point of view, if you look at H1, besides this little folded domain, it has these large disordered regions, and if you look at the sequence, again, you can see immediately it's full of lysines. It ends up with a net charge of +53. So we have these two highly charged proteins, and the question that we started out with was, how do these two proteins interact? First question is, how strongly do they interact, right? And so this is a little single molecule experiment that we can use to measure the affinity. So here we would start with this negatively charged IEP, Prothymosin alpha, label it with a donor dye and an acceptor dye. Here, if we just look at this protein, we get a single transfer efficiency peak. The transfer efficiency tells us that this protein is quite expanded. If you not surprising because it's so highly negatively charged, right? So there's charged with collagen, an expanded, unfolded, disordered protein. And then if you add a little bit of unlabeled histone H1, you can see here a second population coming up where now prothymosin is bound to H1. And you can see here, when it binds to H1, the transfer efficiency increases, which tells us the chain becomes a little more compact. Again, it makes sense because now it binds to this positively charged protein, screens the charges, and so it becomes more compact. And so we can titrate this interaction, and you can see here picomolar concentrations of H1 are enough to form this complex, so that already tells us we have picomolar affinities between these two proteins at physiological ionized strength. Now, tight interaction, but the surprise came when we looked at this complex with circular dichroism spectroscopy, and in particular with NMR, which of course is one of the key techniques that's been driving the investigation of IBDs. It's really one of the most powerful techniques that you can use in this context. And what becomes very clear from these experiments that were done in Biennial lab in Copenhagen, if you look at prothymosin alpha alone, it shows the classical signature of IEPs if you look at these HSQC spectra. And so Harald showed you these spectra where these peaks are highly dispersed, and this is what you usually see when you have a properly folded structure. In contrast, if you have an IEP, if you look at the dispersion along the proton axis here, you see very little dispersion. And so this narrow range is the characteristic of an intrinsically disordered protein. And what's remarkable now is if you add H1, this dispersion doesn't change. You can look at more of the details of the NMR, you can look at secondary chemical shift changes and so on. And all of this tells us that there is no significant extent of secondary or tertiary structure formation when these two proteins bind to each other. Right? So we have very high affinity disordered complex of two proteins, and if they're disordered, that also implies they're still highly dynamic. And again, this is something we can measure very nicely with single molecule spectroscopy. We can look at these long-range dynamics using this method that I briefly showed you, this nanosecond FCS. And again here, we see this approximately 100 nanosecond relaxation time that tells us even the complex, these molecules are freely moving, rapidly moving around. Now, it's a little challenging to get a structure from this now, right? So how can we still get a picture, a model of what's going on? And so to do this, we turn to simulations. This is a collaboration with Robert Best at the NIH, one of the leading people in modeling intrinsically disordered proteins. And so he suggested to use a relatively simple coarse-grained model, and this is the potential function that's being used. I will not go into the details. Let me just mention one point, which is that there's a single free parameter that's adjustable through this model. And so what you can do now is you run simulations of the system based on this simple model, and then you calculate transfer efficiencies from the model. You can compare those to experiment, and if you do that, you find that you can get pretty good agreement between the experimental data for many different pairs of distances that we probed in these molecules, and we means in this case in particular Alessandra and Madeline Borgia when they were in the lab. And so they looked at 28 different distances within and between these proteins in the complex, and if you compare the measured transfer efficiencies, which are the squares, with the simulations, you see you can get a pretty reasonable description even with such a simple model. And based on this model, this is our current picture of what this complex looks like. Right? So you have the red prothymosin alpha, the negatively charged IBD, interacting with histone H1, the positively charged IBD, and they remain highly dynamic in the complex. Here there is this little folded domain of H1. From this you might get the impression that it's very important for the interaction, but it just sort of always ends up in the middle because it has these two disordered regions on both sides that actually delete the folded domain. The affinity is still picomolar. So the main interactions are between the charged disordered parts of these proteins. Nice thing about these simulations is that even predict the chemical shift changes that we see in NMR, at least relatively speaking, remarkably well. So this was the situation in 2018, and just to put this into context, we now know that there's a whole range of disorder in protein complexes. And here on the very left, you have the classical textbook example of two well-folded proteins interacting with each other. And then you can have varying degrees of disorder, and only some of the people that contributed to this knowledge are listed here. Again, the majority from NMR, which really has been a fantastic technique to work on IDPs. And this would sort of be the upper limit in terms of disorder that you can get. And this is not the only example. There have been several examples reported since, both for protein-protein interactions, and also you can imagine a very similar type of interaction with positively charged disordered proteins interacting with nucleic acids as a negatively charged protein. Yeah? So these interactions happen, which is interesting per se, but of course the next interesting question is, how do they differ from the interactions that we're all used to and love? Right? So I would like to start with thermodynamics. Okay, so you might think, oh my God, thermodynamics. It's a more boring thing to think about, right? But I will tell you that thermodynamics actually can tell us something interesting about the mechanism of these kinds of interactions. Does this really have any biological implications? Right? And so, is there any relevance for biological function? And so, I would like to tell you a little bit about this, and let me go back to the picture that I showed you before. And so, to probe the functional relevance, let's look at the interaction of histone H1 at the site of action here on the nucleosome. And it actually turns out that prothymosin alpha has previously been classified as a histone chaperone. So there have been experiments even in cells that showed that increasing the concentration of prothymosin in cells increases the mobility of H1 in the nucleus. So clearly, prothymosin modulates the interaction of H1 with the nucleosome, but it's not being clear how. And so Peter Heideson at the lab decided to study this kind of process using single molecule spectroscopy. And so of course you can come up with different ways of attacking this. And so you can, for instance, attach fluorophores here to the ends of the linker DNA, right? And then when H1 binds, you expect these two linker arms coming closer together. That should change the transfer efficiency, and you can then follow this reaction. Of course, you can also place the fluorophores into different positions. For instance, one fluorophore on H1 and one on the nucleosome, and monitor intermolecular effect. Right? That's also things that we did, that Peter did. And so the first thing that he wanted to do was to measure the affinity of H1 to the nucleosome, because it turns out that that is a very difficult thing to do because the affinity is so high. And so what he had to do in the end was to measure the KD here over a range of ionic strengths outside of the physiological strength range, because here the affinities get so high, it's extremely difficult to measure. But if he extrapolates them into the physiological range, we get femtomolar affinities. And so this H1 really binds to the nucleosome extremely tightly, which is an interesting observation by itself, but it also has an important biological implication, because that means if you now have to wait for H1 to spontaneously dissociate, it would take a very long time. Right? So the question is, how do we get to biologically useful timescales? And so clearly here, other factors in the cell, like these histone chaperones, are likely to play an important role. And so to investigate that question, Peter did a variant of this experiment where he now immobilized the nucleosomes on surfaces. Right? So here now the nucleosomes sit on the surface, and we can focus our laser beam onto one of these molecules and look at it over extended periods of time. And so, for instance, you can monitor the binding of H1 to the nucleosome. It turns out that this binding is very simple, at least in terms of the kinetics that we observe. It's a simple two-state kinetic mechanism. I don't have the time to show you today, but if you're interested, I can show you later on. The key question here was, what happens to this interaction if you add our histone chaperone, or prothymosin alpha? What does it do to this interaction? So what Peter did is he looked at the binding of H1 to the nucleosome, so we had a little bit of H1 in solution, and then he added increasing amounts of prothymosin alpha. Now in the micromolar range, you can see these kinetics clearly change pretty strongly. And if you could quickly think about what's the possible mechanisms here, this would be the simplest mechanism that we can think of, where H1 can now either bind to the nucleosome or it can bind to prothymosin. If this was all there is, this is the kinetics we would expect, right? We would expect the off rate to be independent of prothymosin concentration, because H1 would first have to dissociate before it binds to prothymosin, and the on rate would have to drop substantially, because the more prothymosin you add, the more you scavenge H1, so it's no longer available. But this is what Peter measured, so clearly that's not an agreement. And so this kind of, especially this increase in the off rate as a function of concentration of prothymosin is sort of a classical indicator for ternary complexes forming. So ternary complex in this case, I think you might agree that the most intuitive thing that can happen is, since H1 is disordered when it's bound to the nucleosome, that it can still interact with prothymosin while it sits on the nucleosome. And indeed, we can describe the data very well in this way. We can also recapitulate this using simulations. So here you see H1 sitting on the nucleosome with its disordered pod interacting with the DNA. Prothymosin comes in, starts interacting, so it neutralizes these charge interactions with H1 with the DNA. The simulations done by Davide Mercadante in the lab in close collaboration with Robert Best, and ultimately then the two guys come off. Right? So prothymosin can invade this complex apparently while H1 is still bound to the nucleosome, and this way accelerate dissociation and act as an H1 chaperone. Now, my time is up, and with that, let me just briefly summarize. I started out with this complex of these two highly disordered proteins, and I told you that the thermodynamics are worth paying attention to in these kinds of systems. I also showed you that this may have important functional consequences, and again, there's many of these charge interactions going on, especially in the nucleus. So I think intricate interplay between charged disordered interactions and nucleic acids in the nucleus is really just something we're starting to understand. And then finally, also phase separation is a topic that we've been able to address with single molecule spectroscopy. And that was only possible because of a fantastic team. Alessandro and Madeline got all of the H1 prothymosin work started in the lab. Andrea looked at a lot of the kinetics with some help from both of these Andreas. Maritra looked at the thermodynamics that I showed you. Peter did the nucleosome experiments. Davide did the simulations. All of the simulations are in collaboration with Robert Best at the NIH. And Nicola and Milos are working on the phase separation. Daniel is a key person for data analysis and all the like. NMR, close collaboration with Birgit Gavelund in a joint grant, and Beat and Sinan helped us with the nucleosomes. Thank you very much.